It is my pleasure to introduce our first speaker, Dr. Susan Bailey. Dr. Bailey is a professor of radiation and cancer biology in the Department of Environmental and Radiobiological Health Sciences at Colorado State University in Fort Collins. She serves as director of the university program of research and scholarly excellence in cancer biology and comparative oncology. She is also director of the Telomere and Telomerase laboratory. Dr. Bailey is past president of the Radiation Society and serves on numerous national and international committees. She has been recognized as an Inventor of the Year for her work in technology development and commercialization with the Colorado-based company Chromatid Incorporated. Dr. Bailey is a native of Los Alamos, New Mexico, and received her master's and doctor's degrees in biomedical sciences from the University of New Mexico School of Medicine. Dr. Bailey is a radiation cytogeneticist with particular interest in telomeres and DNA repair. Her research has resulted in over 70 peer-reviewed manuscripts. She has recently demonstrated the value of assessing telomere length dynamics and DNA damage as informative biomarkers of life stress exposures in extreme environments, including NASA's first-year, one-year mission aboard the International Space Station as one of the investigators in the NASA Twin Study. It is my pleasure to present Dr. Bailey.
Well, thank you very much for the invitation and for the introduction. It's really a pleasure to be with you today and talk about living in space. I mean, we're certainly living in very exciting times for the space program as we really witness a new era of human space exploration with our commercial rockets and vehicles like the SpaceX Falcon 9 and Crew Dragon, launching NASA astronauts and even private citizens to the stars. This month actually promises the launch of NASA's Artemis 1 moon mission with the first uncrewed test flight of the new SLS mega rocket and Orion spacecraft that will return us to the moon. As we look ahead to the success of future exploration missions, it's more important than ever to better understand the effects of long-duration spaceflight on human health and performance. Space represents a unique lifestyle and an extreme environment fraught with extraordinary challenges and hazards, which include chronic exposure to the space radiation environment. But first, and before we go there, I'd like to take a few moments just to remind you what chromosomes are and why they're so important as when we think about some of these environmental radiation exposures. So inside the nucleus of the cells of our body, we find about 6 feet of DNA in each nucleus wound up and condensed into very, very characteristic sizes and shapes of chromosomes, which you see here. And they are connected by a centromere that holds the two sides together, the two chromatids. And these very long DNA molecules are capped by telomeres. And we'll be talking about these as we go along. The other important thing to recognize and realize is that ionizing radiation exposure breaks DNA, or so it breaks chromosomes. And these ends can actually get back together incorrectly. So chromosome rearrangements or chromosome aberrations can actually serve as very informative biomarkers of radiation exposure. We also know that stable structural chromosome aberrations or variants are associated with virtually all cancers that have been reported to date. As far as radiation exposure goes, they're dicentric. So two chromosomes that have been broken and gotten back together, each with a centromere, are actually a marker of radiation exposure and have been used for many years as the gold standard really for biodosimetry. There are other types of chromosome biomarkers or aberrations, translocations, which are rearrangements between chromosomes, as well as inversions. And these are rearrangements that occur within a chromosome itself. So two breaks, a piece comes out, it inverts and inserts itself back into the chromosome to maintain polarity. So these inversions, for example, are very much more difficult to detect. So they have been much less appreciated to date, but we will talk a lot about inversions. And another reason that inversions are important is that they have also been reported for radiation therapy associated second cancers have been proposed to have signatures that include small deletions and inversions. So increased frequencies of these types of rearrangements are really characteristic of radiation-induced tumors. So the bottom line really is that we come to this and propose that inversions, in fact, are very important biomarkers of ionizing radiation-induced damage. And they can be so in a variety of applications and a variety of populations, exposed populations. And you can do some of these analyses both retrospectively as well as prospectively. So I'm going to spend just a few minutes talking about a couple of the studies that we've done that are relevant for environmental radiation exposure. And here we evaluated DNA damage and stress in wild boar chronically exposed to environmental radiation from the Fukushima Daiichi nuclear power plant accident, which occurred in 2011. And our study was conducted approximately 10 years after the accident. There were still exclusion zones. People were not re-inhabiting these high dose areas. And of course, over that period of time, several generations of wild boar were existing and actually thriving in some of these areas. So we looked at DNA damage, we looked at chromosome aberrations, we looked at dicentrics. So here you can see in the not only a wild boar, but also what wild boar chromosomes look like. In blue, that's staining the DNA essentially of the chromosomes. The green dots are the centromeres. So here at the arrow, you have a dicentric, 2 centromeres in this chromosome, and the ends of the chromosomes or the telomeres are lit up in red. And when we analyzed or evaluated dicentrics in these wild boar in blood samples from the boar, we did not see any significant relationship with dose, with radiation dose at time of capture or lifetime dose. We also didn't see any significant relationships with telomere length and dose. The one thing that we did observe was that there were significantly lower levels of cortisol, which is a hormone and marker of stress in wild boar that were associated with higher radiation dose rates at time of capture. So basically, the boar that were living in the high radiation zones in the exclusion zone had lower levels of stress, which really makes a lot of sense because there weren't any humans there. And it would be very consistent with what they're seeing happen in Chernobyl as well, that wildlife populations are really thriving in some of these areas where the humans are still not inhabiting the area. We also, as we started to move then toward more human populations, exposed populations, we also wanted to really improve detection of inversions. So here you can see in this study that on the left-hand side, you see what is called MFISH, so a multicolor fluorescence in situ hybridization or FISH of a human karyotype. All the chromosomes colored their own color, and it becomes very easy to identify which chromosome is which, as well as at the bottom here, you can see easy to identify translocation. So one piece of one chromosome, the purple going over here to the green and the green going back to the purple. So we have a reciprocal translocation, fairly easy to identify. We combined that with what we call directional genomic hybridization, what you see on the right. And here we're just painting one side or one chromatid of the chromosome. And in red, you can see the color along here at the arrows. And here, now we can also still pick up that translocation, a piece of it up here at the top. But we can also pick up an inversion at the green arrow, just as a flip of the signal over to the other side. So it's really important concept of this strand-specific methodology, that it becomes much, much easier to see even very small inversions. This particular study, we compared all of this to, or combined it as well with sequencing, with next-gen sequencing, to see if sequencing could identify the breakpoints. It was fairly successful in identifying breakpoints for the translocations, but not of the inversions. So I know there are a lot of advances being made with sequencing, but at least for the time being, it really highlights the importance of molecular cytogenetics and how valuable that contribution can be when we're discovering some of these structural variants, such as those that are induced by ionizing radiation. I'll just give you a couple more pictures because the methodology, the technology has definitely advanced since the first go at that with just chromosome 3 was the only chromosome we had a paint to. We now have a single color to all of them, but we have paints to chromosomes 1, 2, and 3. And again, I point out that you can easily see a translocation here in the middle, as well as an inversion of that switch of signal over to the opposite chromatid. And this is essentially the strategy and the paints that we used for the rest of the studies that I'll tell you about today. But while we're here, looking forward, we now have the capability to actually have whole genome coverage of these single-stranded, single-sided paints. DGH screen, you can see the metaphase chromosome with the chromosomes, all of the chromosomes having their own paint, and it's a 5-color assay. So we're able to, again, very easily identify chromosomes based on their size and color. And as you see in chromosomes 1 and 2 up here, readily identify inversions. So we're looking forward to this as for some of our future studies. Now, we did a very interesting example of a human population, an exposed human population, was part of the 1 million person study. And here we were able to look at a cohort of World War II atomic veterans. So these were individuals that, weathermen, most of them, in the Marshall Islands after doing nuclear weapons testing in the 1950s. And they were accidentally exposed to a plume. The wind blew a different direction than what they anticipated. There were also some of the individuals worked at the Nevada test site during the 1950s. But all in all, we collected peripheral blood from 12 exposed veterans and 12 age-matched veteran controls. All were over 80. There was one that was even over 100 years old. We evaluated chromosome aberrations using DGH. So we were able to look at translocations and inversions and compare those to historical personal monitoring and dose reconstruction that had been done in the past, and to really try to make the very best possible estimate of dose. We did do a standard calibration curve from 6 healthy male volunteers in their mid-20s that really reflected the age at which the veterans had been exposed. And some of the takeaways, the important things that we found is that, in fact, chromosomal signatures of radiation exposure remained for more than 60 years since the time of the exposure. And in fact, this chromosome aberration-based retrospective biodosimetry gave very consistent dose estimates in the majority of cases. And what we also found was that when we combined the translocation and inversion frequencies, it improved the dose estimations even further. So really a very reliable signature of previous environmental radiation exposures. Just a couple of highlights. Inversions occurred at higher background frequencies than translocations, and their frequencies also increased with age and smoking status. Inversions also increase at a higher rate per unit dose than translocations. And I'll just very quickly show you the data supporting those statements. Here we see translocations across the bottom with the blue circles, increasing with dose. And then we also see inversions, the red squares, increasing with dose, but at a higher rate. And you can also see that the background frequencies of inversions are higher as well. Here is just the influence of age on the left, where you see that like translocations, and even more so, inversions increase with age in the young adults compared to the over 80-year-old cohort. And for smoking, here at the dotted line is just the average non-smoker rate. And again, you can see that both inversions and translocations increase frequency with smoking status as well. So now I think we're ready to go ahead and jump off the planet and start looking at life on board the International Space Station, where in low Earth orbit, about 250 miles above the Earth, and astronauts living in space for extended periods of time, either 6 months or 1 year are pretty much the standards. But they are chronically exposed to the space radiation environment. So I'd like to also just thank some of the participants of the study that I'll be telling you about. You see here on the left in the front is Kjell Lindgren, who is a NASA astronaut who will be returning to space this year sometime. Behind him is Kimiya Yui, astronaut with the Japanese Space Agency. And on the right in the front, you see a gentleman that you may well recognize as Scott Kelly, because he was in fact the astronaut selected by NASA for their first one-year mission, which was flown. He flew in 2015 to 2016. And certainly a lot of PR around that event. But interestingly, the most of it was focused around the fact that he had a twin. But first, you have to realize that there was a one-year mission as well. And this was a lot of very real science that went into this, many collections being made a year before the flight, a year during the flight, as well as up to a year after his return. Lots and lots of different studies going on associated with the one-year mission. But as I mentioned, he does have an identical twin brother. And it was interesting that it was Scott Kelly himself who brought up at a press conference, well, are you doing anything? Because, I have an identical twin brother. He was also a former Navy test pilot and astronaut, a pilot, commanding the space shuttle. So very similar in nature and nurture, and really the perfect experiment. And so NASA very quickly put together what became known as the twin study, and they selected 10 different investigations from around the country. And it was really the first time that they went into or ventured into the omics world. So genomics, DNA sequencing, RNA sequencing, almost every omics you can think about, proteomics, metabolomics, and as well as their other more standard like physiology and biochemical profiles and cognition were all included in the twin study. And I'm just going to very briefly highlight some of these studies so that to give you an idea of what all was done, but also how many other real firsts were achieved during the twin study. So here we see Andy Feinberg and his team at Johns Hopkins University, who was looking at epigenetics or the epigenome, doing DNA sequencing and RNA sequencing. And it's one of the basic things to any of these molecular studies is the fact that you have to be able to pipette, you have to be able to move liquid from tube to tube. And that turns out to be quite a challenge in microgravity. So this team was actually involved in doing that. They were on what they call the Vomit Comet parabolic flight that simulates microgravity for short periods of time, and trying to work out how they could actually do pipetting in space. And they worked that out and actually paved the way for the very first DNA sequencing in space, which Kate Rubins did in 2016, and is now almost routine. Of course, nothing in space is routine, but they do that quite regularly now on the ISS. Chris Mason and his team were also looking at DNA and RNA methylation, doing a lot of sequencing, looking at epigenetics, and they too were really very instrumental in getting some of these really sophisticated sequencing experiments accomplished, and doing so during flight. So that's another just really amazing study. And again, most of these things that they were measuring, so gene expression, how genes are being turned on and off during spaceflight, the vast majority of all of them returned to near baseline by the end of the study, so 6 to 9 months out after his return. Scott Smith and his team at NASA were involved in doing the biochemical profiles, so really just standard blood and urine collections, and monitoring a variety of analytes that reflected diet and exercise, weightlessness. Certainly, the diets on the space station or during space flight are very, very different. They're very defined. They're kind of boring. And many of the crew members complain, that it just gets old. So many of them lose weight. In fact, Scott lost about 15 pounds for the year that he was up there. And the other thing is that, you have to realize that, growing your own food is really going to be very important for future missions, a long duration, when they get further and further away from the Earth. And in fact, they did grow some lettuce on the space station during his time up there. And on the bottom here, you see Scott Kelly and Kjell Lindgren eating the first space salad. So the first time that they'd actually grown lettuce on station. Emmanuel Mignot and his team looked at the immune response. Certainly there is a real inflammatory response when astronauts return to Earth, and that is very common and observed in almost all of the astronauts, including Scott. But the first for this study was the first time that a vaccine has actually been tested in space. So on the bottom here, you see Scott and Mark each giving themselves a flu vaccine. And in fact, the response was very similar in both of them, whether they were in space or on the Earth. And that's the first time that has ever been done. Stuart Lee and his team at NASA did some metabolomics looking for cardiovascular effects or biomarkers of cardiovascular effects, artery structure and function during spaceflight. You can see some of the different equipment that they have on board, that they're able to monitor some of these things actually during spaceflight. And again, most of these endpoints that they analyzed returned to baseline within about 6 months of Scott Kelly's return. Brenda Rana and her team at University of California, San Diego looked at proteomics, and they so the proteins involved in what they refer to as fluid shifts. So on Earth, we have gravity pulling on us and all the fluids in our bodies. So it keeps a lot of things more toward our feet and our legs. In space, in microgravity, you don't have that. So everything goes up. And so these fluid shifts then often cause astronauts, why they look puffy sometimes when you see, their videos in space or on the space station. It can affect their vision. It can increase intracranial pressure and intraocular pressure. And so you see Scott and Kimia Yui doing some of these measurements on the space station. But she was looking for proteomic markers of this phenomenon. So perhaps we can be better at coming up with a countermeasure for it, because it can influence astronauts performance as well as their vision during flight. Matthias Basner and his team at the University of Pennsylvania administered cognitive tests. So they evaluated cognitive performance before, during, and after spaceflight. Very common, they do this with a lot of the astronauts, but now for this study, of course, Mark and Scott were both doing these cognitive tests. And in fact, Scott did have some decline in cognitive performance upon his return. And in fact, if you've ever read his book, he wrote a biography of endurance. And even there, he talks about how he really didn't feel like himself or normal for about 9 months after his return. And that was very consistent with what they found here in this is cognitive performance as well. But by 9 months, he was pretty much back to baseline levels. And this study of the microbiome by Fred Turek and his team at Northwestern was looking at the microbiome. And if you've ever wondered what the outhouse on the International Space Station looks like, here's a picture. It very much reminds me of 2001 Space Odyssey, that this is going to require some instructions. But they did these studies and evaluated the microbiome again during spaceflight and found that, yes, of course, it changes. It changes with the environment in which you're in, but it also reestablished itself upon return to Earth, again, within about 6 months of his return. So I hope I've given you a very quick whirlwind kind of view of the comprehensive nature of the twin study, because it really was at the frontier of personalized medicine approaches for evaluating health effects of individual astronauts. And it's kind of laid the foundation for how these things will be done and how health will be monitored in the future on future missions. But there certainly is little doubt that this pair of twins is the most profiled that there ever has been, either on or off the planet. And it's interesting that they're also one of the most interviewed. They certainly had a lot of interviews, a lot of attention around this mission. And one of the questions that was very often asked was whether or not Scott would return to Earth younger than his brother Mark because of his space travel. Of course, this was often in the context of watching Interstellar or talking about Einstein's twin paradox. But those who have done the calculations assure us that it probably was, if it was anything, it was only a few milliseconds difference, because the space station is in low Earth orbit. It's not traveling at the speed of light away from us or relative to us. So he's not experiencing, time dilation in that sense. But the question of aging associated with long duration spaceflight, particularly as we go further and further and longer, really is an important one. And because it's also associated with all the age-related degenerative conditions that go along with it that could influence astronaut performance. So the question of aging really is where we come into the twin study, where we were proposing that, in fact, the very first time that a biological marker of aging would be evaluated in the astronauts on long duration spaceflight. And what we proposed is that telomere length dynamics, or changes in telomere length over time, not only would be an important marker of aging, but it would also be important because the dynamics of it, or the changes of it over time, would be reflective of all of these different exposures and stresses that the astronauts are under. So it integrates those things. It integrates their genetics, it integrates their lifestyle factors, their exposures, things like not only space radiation, but also microgravity, isolation, the exercise, which they do a lot of because to help battle bone loss and muscle wasting. So all very unique lifestyle factors are being integrated into this biomarker of telomere length. So let's just talk a minute then about, well, what in the world are telomeres? Well, telomeres are in fact just the natural ends of our chromosomes, so linear molecules of DNA. They shorten with cell division, and so they've been proposed as kind of a mitotic clock. They also shorten with a variety of other things like inflammation, oxidative stress, lifestyle factors, as I mentioned, things like nutrition, psychological and physical stresses, and environmental exposures. Air pollution, for example, or even UV and ionizing radiations also can contribute to telomere erosion. Telomerase is just the enzymes that helps to maintain telomere length, but it really is not sufficient in the majority of our cells to do so. So that's why telomeres shorten as we get older. And this idea of telomere length dynamics, then this changes over time or rate of loss of telomere length. How can we, better maintain it, or what is it that's actually accelerating that process, can provide a very informative biomarker of aging and the diseases that go along with it. Things like reduced immune function, loss of fertility, dementias, cardiovascular disease, and cancer. Again, all things that would be very detrimental to astronaut health, particularly during a mission. So telomere length dynamics then really reflects or integrates the interplay between all of these factors. And they can be viewed as sentinels, really, of some of these exposures. So if we think then about some of the exposures, and again, one of the major hazards of long duration space flight is space radiation. And that's because, here on the Earth, we are protected by the Earth's atmosphere and the magnetic field, so that we are not exposed to a lot of the very harmful types of radiation that astronauts will be exposed to once they get outside of low Earth orbit. But even in, even in low Earth orbit, they do get more, about an order of magnitude more exposure than we get here on Earth just from natural background radiation. And certainly shielding is not a very good, or it isn't much of an option, because it's very problematic, because it's usually very heavy. Concrete, lead, water are all very, very heavy and can't be flown. So, and sometimes shielding isn't even an option at all, like on an EVA, one of their extravehicular activities. So here you see Scott Kelly. And actually, if you look real close, you can see in his visor a reflection back of the Earth. So cosmic rays, galactic cosmic rays, emissions from the sun are all very, very serious problems that we have to face as we start making our way toward Mars. And because, as I said, the International Space Station is in low Earth orbit, and there's a long way to go to get us all the way to Mars. And this is just a really interesting and informative slide that shows you a film that was strapped to the ankle of Neil Armstrong, so the first man on the moon. And really the Apollo missions were the very first time and only time that we have gone beyond low Earth orbit and outside, completely outside of the Earth's protection. And you can see here that this film has radiation tracks, so ionizing radiation tracks across it. And the small little blob things are like X-rays and gamma rays. You have kind of right down the center, you've got a proton track, and then going across the other way, you have a much more densely ionizing track of a charged particle, a heavy charged particle, which they call HCE ions. But you can readily appreciate that if you think of the cells that are and the DNA that is being hit by these ionizing tracks of radiation, that it's definitely going to be a problem. This mission, the Apollo 11, was only 8 days. So this is the kind of dose that he was experiencing after only 8 days. So the objectives then of our study, back to the telomere study, was to test the hypothesis that in fact telomere shortening would be accelerated, and that DNA damage responses like chromosome aberrations would be elevated with spaceflight. We wanted then to assess, what we did was we assessed telomere length and these DNA damage responses or chromosome aberrations between the astronauts, so Scott Kelly and his ground control, Mark Kelly, as well as in a second cohort, we were fortunate enough to have a separate study where we had 10 unrelated astronauts and their age and sex matched controls. So that really helped us establish more of the significance of our findings. The unrelated cohort, I'll just mention, although Scott was on a 1-year mission, most of our other astronauts were only on 6-month missions. But by having these 2 very important cohorts of astronauts, we were able to make some important comparisons. We could compare telomere length and chromosome aberrations between the twins to identify spaceflight specific factors or non-genetic factors that influence telomere length. And we could also make comparisons with and between the unrelated astronaut cohorts and their controls to really evaluate the role of genetics or individual differences and susceptibilities to some of the changes. The other really important thing, and I've mentioned it a few times, but it's really important that this was all of these were longitudinal studies. So we could evaluate these changes over time. I get at this idea of the dynamics of these processes. So blood samples were taken, and we did our assays all in blood. Blood samples were taken before, during, and after spaceflight. Really very straightforward and simple experimental design that was much more challenging in execution and actually getting these samples from these very, very busy people who were, really immersed in training around the world before their flights. And then the samples that we got in flight were even more challenging, where the blood samples were actually taken on station. You see down here a couple of the astronauts, they help each other do the blood draws, and they would do that right before the Soyuz would return a crew, and they'd return those blood samples just under ambient conditions back to Earth, and then jet them to Johnson Space Center, where we had them in the lab within about 48 hours of draw. So really a remarkable feat to get those samples back to the Earth in time for us to do our analysis. And then post-flight, we had a number of post-flight time points as well, going out as long as 9 months to a year. Again, and these can be challenging because oftentimes the first thing they do, just as Scott Kelly did, was retire. So it can be challenging to follow up on them, but really a remarkable opportunity to get these samples, and then to have the power of, with the twin study, of not just only having our observations of telomere length and chromosome aberrations, but also being able to correlate that with all of these other studies. And we could ask questions like, well, what about inflammation? What about oxidative stress? What about gene expression? What can you tell me about some of these other things that are going on? So it really became very, very powerful and important mechanistically. So let's go ahead and I'll just quickly run through some of the data so you can see what we found. So this is telomere length, average telomere length determined from DNA by a qPCR reaction, and it's for the twins. And as we go through this, you'll see that Mark Kelly, the ground control, is always in green, and Scott Kelly, our flight subject, is in blue. And again, we've got pre-flight, mid-flight, post-flight values for telomere length. And you can see that Mark Kelly, again in green, is pretty stable throughout the study. Whereas Scott Kelly in blue, you see that in fact, during spaceflight, mid-flight, his telomere length was actually longer than it was before or after. So exactly the opposite of what we hypothesized would happen. So that was really a very unexpected and surprising finding that we were very anxious to follow up on. And so we did have a number of other crew members that we had in-flight samples for. And so when we looked at the entire cohort again. Now we're looking at a cohort of 11 individuals. We do see, in fact, the significant increase in telomere length during flight, and then a significant decrease in telomere length compared to where they started with or to the controls post-flight. So overall trends, astronauts had shorter telomeres than their healthy age-matched controls at baseline. I've already mentioned the significant increase in telomere length during flight. And then at the end of the day, overall telomere length was significantly shorter after space flight than it was before. We also did another method of telomere length measurement, a cell-by-cell type of approach that we call Telomere Fish or Telofish. It's just, again, fluorescence in situ hybridization, but this time with a telomere probe. And here you see it on metaphase chromosomes lighting the ends of the chromosomes up, or the telomeres, with pink, so we can very easily see them, and we can very easily measure fluorescence intensity, so that we know, and so the brighter the telomere, or the brighter the signal, the longer the telomere. So it's a very straightforward way to do that, and it also enabled our evaluation of chromosome aberrations. And when we did our telomere length, or telofish, on the twins, again, we saw a very consistent or similar trends, where Mark Kelly was fairly stable, but we see this increase in Scott Kelly of telomere length during flight. If we look again at all the astronauts, it becomes much more significant and obvious that, again, astronauts seem to have shorter telomeres than the controls. The astronauts' telomere length during space flight is significantly longer, and then after space flight, it's significantly shorter than what they started with. So all of this was very, very consistent, and essentially validated our PCR results and gave us a lot more confidence in the results. The other really important thing that we can do with the telofish data, because it gives us thousands and thousands of individual telomere lengths, we can generate these telomere length distributions. So you see along the top here, this is for Mark Kelly. The yellow are the shortest telomeres, and then progressing then to the red, which are the longest telomeres. And so you can actually see these shifts in these populations of short and long telomeres. For Mark Kelly, he was, again, pretty stable, everything, very consistent with what we've seen. There weren't a lot of shifts. If you look across the bottom, this is Scott Kelly. And again, you can see quite a different story, that there are some fairly dramatic shifts. You definitely see mid-flight, which is the middle panel here, more long telomeres than he had to start with, consistent with what we've been seeing with average telomere length. And then at the end of the day, by the end of the study or post-flight, you see a really dramatic increase in the number of short telomeres and very many fewer long telomeres. So again, just all very consistent with what we were observing, and supportive of it as well. And if I still haven't convinced you, we also, in collaboration with a couple of the other twins investigators, across the top there, Chris Mason and his group did some nanopore sequencing of telomere length, so long reads of telomere length. And again, you can see the very same thing. Here's Scott Kelly. In mid-flight, he has more long telomeres than he did before or after, whereas Mark Kelly over on the right is, again, very, very stable throughout the study. Kumar Sharma was able to, they had urine samples, and he did telomere length and urine by qPCR. And again, very striking and very significant increase in telomere length during spaceflight. So at the end of the day, we were very, very convinced by the results for telomere length. But now what about chromosome aberrations? What was happening with chromosome aberrations? Again, thinking about the space radiation exposure that Scott in particular was experiencing. So here we have the twins again. We have inversions in the solid bars, and you can see that, or I should start with translocations in the stripe bars. You can see that they're pretty similar with Mark and Scott's pre-flight. They increase during flight, which is exactly what you would expect because of the radiation exposure. And then they start to drop off post-flight because many of them will be lost. Those cells will die. If we look at inversions, it's very much the same, certainly higher levels, again, as we would expect and as we've seen before, and fairly similar between Mark and Scott at baseline. But what happens is that indeed inversions are elevated during spaceflight, and they remain elevated after spaceflight. So out as far as 9 months afterwards, he still has elevated levels of inversions. And that, of course, is because a lot of these can survive. They're small inversions and they don't kill cells. So the consequence there could be that there is an ongoing kind of instability, or there's damage to stem cells that could be of a concern in years to come. If we look, then let's do the same thing here. Let's look at everybody. Let's look at the entire cohort from whom we had in-flight samples. And if we look across the bottom, let's look across the bottom first. You can see, again, we saw very few dicentrics during spaceflight, but there were a few. There were a few translocations, but inversions, again, it's pre-flight, mid-flight, post-flight. Pink, there's a significant increase in inversions across the board, and they remain elevated after spaceflight. The other interesting thing that we observed that was very spaceflight specific were what they call satellite associations, and I'll show you a picture of that here in a moment. But these have been associated with low dose radiation exposures before as well, in hospital workers, for example. So that was an interesting finding. Across the top, we have some other events, more recombination related, but again, seem to be occurring during spaceflight. And this one in particular, this heterogeneous sister telomere phenotype, again, which I'll show you in a moment, was very spaceflight specific, and I think is giving us some clues as to what might be going on. So here, let's just take a look at them so you can appreciate what they look like. So on the left, again, there are these heterogeneous sister telomeres. So the two telomeres obviously are very, very different size. These have been proposed as a hallmark of a very different pathway of telomere length maintenance that does not require telomerase, an alternative type of pathway that can be activated by chronic exposure to oxidative stress. On the right-hand side, you see one of these satellite associations. So there are the chromosomes that have very short P-arms, and the satellite DNA seems to associate with one another. And as I mentioned, that's actually been proposed before too as a biomarker of chronic low dose radiation exposure. So these were both very exciting findings, and we're pursuing both of them as to what they might be telling us about what's going on, what the mechanism behind that is. And certainly there's a lot more to the story than what I've been able to tell you here. If you're interested in either the original twin study, which came out in Science, or the end of 2020 and beginning of 2021, there was a whole series of about 30 different manuscripts that came out across Cell Press. I would refer you to those for the rest of the story. But to summarize our findings just very quickly here at the end, that indeed the twins started off with very similar telomere lengths as we would have expected. Astronauts in general tended to have shorter telomeres than the controls, and they had significantly longer telomeres during spaceflight, and just completely opposite of what we imagined. Telomere length also shortened very, very rapidly upon return to Earth. For Scott Kelly, it happened, we had samples within 48 hours, and they were already quite a bit shorter by that time. And then they stabilize over the coming months, but there's still these really dramatic shifts in populations of short and long telomeres, and they have many more short telomeres after spaceflight than they did before. And that's really where disease risk or aging risk might play a part. And some of our cytogenetic observations certainly have given us some mechanistic insight, as have the other twin study results. So we really are becoming convinced that it's radiation, chronic exposure to the radiation space environment, these low dose rate exposures could be responsible for what we're seeing, certainly responsible for the DNA damage responses, the inversions and the satellite associations that we observed during spaceflight. We also know that there's mitochondrial dysregulation and chronic oxidative stress during spaceflight. And so there's this ongoing damage to telomeres, and there's replication stress that I think, again, could really be contributing to the telomere phenotypes that we have been observing. And we also saw, I'll just quickly mention, we had some twins that were climbing Mount Everest and saw a somewhat similar extreme environment, and saw some very similar types of phenotypes with the telomeres. We also saw increased cell killing, which could be reflective of lymphocyte radiosensitivity and reduced white blood counts. So again, here, what we're thinking is that it could be that we're seeing a redistribution of some of these subsets of cells associated with spaceflight, much like we've seen in some of our radiation therapy patients. So again, it could be that it's not telomere lengthening per se, it's not actually adding on telomere length, but we're looking at different populations of cells. So that's currently what we're following up on and looking forward to testing more as we go forward with the 1-year mission project and what they're now calling CIPHER. But that's kind of the direction, certainly, that we're going. And you might, by this point, be asking yourself, well, why should we even care about this? Of course, it's important for spaceflight, but it's important for us as well. We all care about aging and improving or maintaining our health span. And although Scott Kelly came back from a year in space briefly taller and 15 pounds lighter and maybe even a few milliseconds younger, going to space really isn't a fountain of youth. And we've highlighted that in a recent article in this Science of Aging journal. But I think it's still important for us to think of aging in the larger context, or really expand our vision of aging as we think about space. And we can view space really as the next frontier of aging studies and potential breakthroughs, because that's what it's going to take. It's going to be essential for us to do that, to ensure the success of future long duration missions, particularly as the number and diversity of space travelers and even space tourists increase in the very near future. We're going to be seeing that happening. In fact, we just recently witnessed the first all-civilian mission to orbit, the Inspiration 4 crew, who was in orbit for 3 days. And we've even seen Captain Kirk blasting off to space as the oldest person to fly in space. But he was only up for about 3 minutes officially in space. But there's just no doubt that it's an exciting time for Trekkies and Earthlings alike to go where none have gone before and to live long and prosper while they're doing so. NASA and its commercial partners are really rapidly advancing innovative space technologies, and with the Artemis team of astronauts, plan to send the first woman and the next man to the moon, even before that, by the end of this decade. So with that, I'll stop, and I, of course, have to thank NASA for their support and just vision for this entire project, and all of the people really here at CSU, as well as Weill Cornell Medicine and UT Health San Antonio, that contributed to the studies I showed you. And of course, a big thank you to the twins investigators, a remarkable group of people that it was a real privilege and pleasure to serve with along this really amazing journey. So with that, I'll encourage you to stay tuned and buckle up, because we are going. Thank you.